That's good. Well, you're welcome.
I can't
You can't
hear you, Angela.
Basically, the comment was that when I address you for the first couple of go-rounds, identify yourself by name, because we would not be able to tell that it's a female on a telephone connection otherwise. So just, so we wouldn't be able to identify you. All right, let me set up a couple of ground rules. You know that the discussion is focused on peroxide, and this is probably the first roundtable that we've had on peroxide in, as far back as I could remember, which goes back a fair amount. And so it's don't laugh, Chris. So it's an exciting opportunity.
I know.
It's an exciting opportunity, and I think it gives us an opportunity to talk about a lot of new things, and I really want to focus on the science. One of the things I want to do is make sure we don't trash multi-purpose solutions. Traditionally, the evolution of lens care has been multi-purpose solutions versus peroxide. I really don't want to go in that direction. The two are synergistic and there are different uses for each. So I want to really focus on that. And I really want to focus mostly on the science. So I think the first question, and I think, let's see, who should I start with? Chris, why don't we start with you? I know you've done all the reading, because you read through all of the articles, which is actually very impressive to me. Talk about a Fenton reaction and how. No, actually,
that's an interesting one.
Just kidding. Let's talk about actually how peroxide works. I know we use it, but actually, how does it work?
Well, peroxide is a nice disinfecting agent for lots of things in the body, not just eyes. And it was determined a number of years ago that using a contact lens system that utilizes hydrogen peroxide, starting at a 3% concentration level, does a very nice job disinfecting the microbes and bacteria and pathogens that can get on a lens during a wearing cycle. The challenge for a peroxide solution is neutralizing it in a reasonable period of time so the patient can then reinsert the lens the next day without causing any compromise to the ocular surface. And fortunately for contact lens wearers, that science has come to fruition for a number of years, and we've had a lot of happy patients who have been relatively symptom-free for a number of years and complication-free.
That actually sounds reasonable. Was that you, Chris? Okay, wait. Ladies first. Chris?
Okay, so peroxide is a really interesting chemical, really, because we all think of peroxide as being very benign, because we think of it as breaking down to water and oxygen, and that those are very benign things. But peroxide is a very potent and very powerful chemical disinfectant, and it works by producing hydroxyl-free radicals that attack the lipid membrane. It also attacks the DNA, it attacks the mitochondria and other cell components. So hydrogen peroxide can be incredibly destructive, even with these free radicals being very short-lived. They're highly reactive but very short-lived. And because hydrogen peroxide is lipid soluble, when it comes into contact with a cell, it can easily penetrate into that cell and then sort of release its cascade of what happens from there of destruction inside of the cell. We are surrounded by peroxides all the time. It's in the food we eat, it's in the water we drink, it's in the air we breathe. So our bodies are used to peroxides. And our bodies have come up with a defense mechanism for handling peroxides, and that's primarily catalase, although there are some other antioxidant enzymes that protect the cell from the metabolically produced hydrogen peroxide. When it becomes a disinfectant is when the cells or our bodies are overwhelmed by increasing concentrations needed for disinfection. So typically we can buy over-the-counter peroxide, which is going to come in 3 to 6%, which is the 3% is what we use for contact lens disinfecting solutions, at least what's available here and now, although higher concentrations have been proposed in the past. We use higher concentrations of peroxide, like 6 to 25%, as actual chemical sterilants. And that is just such an overwhelming amount that our bodies could not overcome that as an antioxidant. How peroxide works is it can disinfect amoeba and viruses and the protozoa viruses, bacteria, fungi. It's active against all of these different agents. And it's going to depend on contact time or on concentration, because things like amoeba and fungi have these defense mechanisms against them, because peroxide is such a common thing in our environment that they've actually built up defense mechanisms to counteract peroxide. So it's all really a factor of contact time and concentration of the peroxide as to how well the peroxide works.
Okay, great. We're done. that was like, all right. But I'll urge everyone. That was really good, and actually you covered really about 8 of my questions. But so Chris has always been an overachiever, by the way. So I'll urge all of you to keep your responses relatively concise and focus on one thing, because we want this to be sort of an interactive roundtable.
You're talking about Chris on the phone.
No, I think she knew. No, I think she knew. No, that's all right. No, that was very good. Bill, you had a quick comment?
Yeah, I think that there are two comments. One, that people do tend to minimize peroxide. We have to realize that actually our body, the bodies in our cells actually use peroxide to kill other cells. But more importantly, a person who's ever had it in their eye will never minimize it again. The fact that you can kill things like acanthamoeba cysts when they're resistant to hours and hours of boiling, which will not affect them, demonstrates how really powerful peroxides are.
Okay, good. No, that's.
Peroxides are used in our body in many positive ways, not just for disinfection. They're used for wound healing. It signals inflammation. So peroxide is actually an inflammatory mediator within our body, and it's up to our body to moderate that. To hold these pro-inflammatory mediators at bay. It causes, it helps with wound healing, angiogenesis, fibroblast creation, wound closing, epidermal growth factor. I mean, it's very important for many processes that actually go on within our body, and it's up to these catalases to keep it at bay.
All right, let's get back to contact lenses, and let's focus on contact lens disinfection particularly. And those are all good points. I think the point that peroxide is produced by the body. It's part of normal metabolism. The body has strategies for managing it, neutralizing it, I think is important, and I think we do need to include some of that science. I think that the fact that it's a natural product is, I think, fairly important. Peroxide came at a time where we didn't have a lot of choices, and I'm thinking who here is old enough to actually remember when peroxide was first introduced by American Optical years ago. I think they were the first peroxide, and I realized that that's me. So I'll just say that when it was first introduced, it really was a significant, almost a godsend.
What year was that?
It was like, thanks, Loretta. It was I think probably
1942.
No. That's when you remember it now. It was like 1982, I think, something in that range, give or take a little bit.
Great, that's as old as I am, Mark.
Yeah. That's the good thing. So we have a wide range of ages on this panel and a lot of experience. So we talked about neutralization, and that's obviously very, very important. And then I want to talk about disinfection, and Loretta, I'm going to tap you certainly for that. Mila, we have a number of neutralization strategies. Can you describe them? What are the differences? What are your clinical preferences?
Yeah, well, from a patient perspective, we always try and in our clinics simplify things as much as possible for people. I like to minimize the amount of steps that they require to do whatever is required to treat any type of disease, to fix vision with contact lenses. And a platinum disc strategy, in my mind, just makes sense because it's simple. Patients don't have to put a lot of thought into it. They simply have to follow the steps, placing the contact lenses in the cage, making sure they fill it up to the line, and then dipping it in there. And they don't have to worry about an additional tablet or something to add to the system to neutralize it. So it just, in my mind, it makes a lot of sense because it's simple, and for the reasons that Dr. Levens, Dr. Sint, Dr. Townsend, and even you mentioned, it's so highly effective in the way that it disinfects that it just makes sense as, in my mind, a problem-solving solution for those people who may have hypersensitivities or other issues where we may feel that peroxide's warranted for these patients.
That's good. I think that's really good and to the point. The other systems using catalase, what issues do they typically have? Chris?
One of the issues is repeatability, in that the tablets don't always agree with one another, and sometimes they neutralize at different rates and different speeds, and that can be problematic to the patient and the patient's eye. Human nature is challenging, as we know, as practicing on human beings, in that one of the benefits of the platinum disc that Mile mentioned is the fact that it's relatively long-acting, and it is very, very consistent. It is not unheard of for a patient to run out of their catalase tablets, and so they may, very much on accident, the very next day, well, I'll go ahead and put this in there and don't have a tablet and see what happens. And their eyes is oftentimes very angry then the next day due to that poor behavior. Whereas with the platinum disc, you really can use it and use it and reuse it and reuse it so often that it really takes some of the compliance issues away, because we are human and we tend to forget things.
Well, that's a great point, because I do recall the days where many patients used catalase tablets and other neutralization strategies where they had to actively be involved, and they would forget. And they'd wake up in the morning and suddenly remember, much to their frustration and sometimes great discomfort. So they didn't forget that often. Yes, Chris?
I was told, in optometry school, that your patients are only going to forget to neutralize their hydrogen peroxide once. They'll only do it once. And now that I've been in practice for 20 years, I have discovered that no, people will do it more than once. So, I think there can be a learning curve to how and when people neutralize. I think one of the other advantages of using the platinum disc is now that it all comes together as a case. As the disc starts to wear off and the patient may notice a little discomfort as soon as they put the lens in, they replace the whole case. So not only does it just neutralize the hydrogen peroxide, it also drives case replacement in these patients as well.
Well, that's a great point. When we went from replacing discs to replacing cases, and the cases are distributed with the solution, so it minimizes that issue, and it also minimizes the issue of biofilms and other contaminants in the bottom of the case, which we'll talk about in just a second. One of the things we sort of led to is the issue of safety, and I know, Loretta, that's obviously something you focused on in a lot of ways. How safe is peroxide?
How safe is it in the patient's eye? If neutralized or
in the patient's hands? Just using it as a system. I mean, do you consider it a very safe system, or is the complexity make it less safe? Or
well, we've all had patients that used it in the wrong manner, perhaps even rinsed their lens with it, or a neighbor was over and used whatever was in their medicine cabinet and put the peroxide in their eye. We've all heard those stories. So in cases of non-compliance, you will get a transient, up to 3-day, probably ocular surface acute toxicity. Never have seen a major problem from that. Of course, it's painful, and we'll get that phone call, and sometimes we don't even treat them anymore. We just talk through them on the phone, but they always get better. So, there's no long-term complications from putting it directly in your eye. But now, if used properly, how safe is it? Well, I've never seen a complication directly attributed to, the peroxide system itself. We can talk about infiltrates. I'm sure you're going to. They still occur with peroxide systems, but of course they're minimized compared to multi-purpose solutions as a group. So it reduces to enough parts per million of residual peroxide that it's not toxic to the eye once it's neutralized and put back in properly. I am younger than you, but old enough to remember the days when you had to remember to let it soak in the 3% peroxide for 10 minutes or 20 minutes, I can't remember the time, and then you had to add the neutralization. So there were many times that patients did forget that step years ago.
No question, it did happen. And, I think there were a lot of people who comment that peroxide is inherently not safe because it requires patient interaction, and frankly, I found it over the years to be as safe as any other product. I think your point is well put. It's as safe as the patient allows it to be. If a patient doesn't pay attention, they can get themselves in trouble. In contrast, practicing for most of my life in New York, I found that a little bit of a sting wasn't that big an issue, and because peroxide neutralizes pretty much whatever it comes in contact with, from a biocidal perspective, they essentially sterilize their eye when they make that mistake. So
I call it a gentle giant.
That's a good way of putting it.
That's how I refer to it sometimes with my patients. It's strong, but it's gentle when it's used properly.
Yeah.
Kind of like Rick Weidbart.
A gentle giant. Well, that's a good way of putting that too, Chris. So since you volunteered, what problems have you seen with peroxide from a technical point of view? Have you seen any? And what solutions have you come up with? Have you come up with any unique solutions in terms of ensuring compliance and making sure patients are following your directions?
Who are you asking?
Kristen.
Okay, well, sometimes peroxide is maybe not appropriate for certain, say, prosthetic lenses. There may be some issues with some laminated lenses, for example. So it isn't necessarily appropriate for every single lens that's on the market, but for the vast majority of what we consider normal and lenses, it works quite well for. Kind of which line you're going for there?
Oh, no. In other words, there's no such thing as a perfect product. Every single product has an issue. And I'm just curious as to whether or not, in fact, Chris, you're nodding, whether you've encountered issues that you've overcome through patient instruction or strategy.
Sure, thanks, Art. I think what's really on the front of the mind of the patient as well as the practitioner is neutralization. I think that's really the biggest question because of the patients that both Laura and Mile mentioned who have accidentally put hydrogen peroxide that's not very well neutralized on their eye, and it's not very comfortable, and it's not great in terms of the way it looks and feels. I think in designing a product for human use and or human consumption, responsibility in that design is very critical and very key. And one of the things that there's been some very nice science looking at is how this translation from 3% hydrogen peroxide gets reduced, so when this lens is put on the eye, a count in parts per million of remaining peroxide. What is it? And I think ClearCare, the design has been very, very responsible in that it's been so low that we really have not encountered a lot of adverse reactions on the eye due to what could be the opposite, where some products can come to market or have been on market where the parts per million is significantly greater than that. And it can be subclinical, it can be clinically obvious, neither one being good for the patient.
That's a great point. We have almost a responsibility to ensure the lowest residual peroxide when the patient uses this product. Loretta, you were going to make a comment.
Well, not on that point, but on the previous point of you asked, what problems have we seen with peroxide?
Okay.
There's definitely patients I avoid peroxide with. One is the occasional user, because as the peroxide gets converted or neutralized, as you said, to relatively non-preserved product, then certain organisms that can survive the cycle, because they, as you guys have mentioned, organisms can produce catalase and peroxidases as well, so they can survive a cycle. And they can grow up over time. So if a patient is a weekly wearer, I don't recommend peroxide because when they go to put that lens in, you might be surprised at the organisms that are found on that lens. So they're better multipurpose users. There's also certainly the patients that we see in our practice that we would never even ask them to use peroxide, and I'm sure you can all name a few. You just worry about their compliance. Even some peroxide users top off. So if you've got the patients that are skimping on products, and you already know that probably peroxide is not the best for them.
Yeah, no, that's a good point. And in fact, non-compliance is not limited to any one system or any one product. And there are certainly patients that you can spot from a mile away once you have a little bit of experience that you treat with kid gloves because you know they're going to not listen to whatever it is that you suggest. Yes, Mila.
Could I just add something to the compliance perspective? I think those are great points too. One of the things we do in our practice, and we try and do this just to minimize any of the adverse events, which is really going to happen if somebody does what they're supposed to do with the peroxide system, it will work as a good system. So what we do is two things. We educate them in the exam room, and that's where I start talking to them about the system and just the fact that it's different, and you're going to have to follow some very specific instructions that are a little bit different than what you might have been using. But that message is actually reinforced before they leave the office with one of my technicians, where they go over very specifically exactly what they're supposed to be doing, and most importantly, they actually leave with a sample of it so they can practice it before it's actually time for them to go to the store shelves and purchase it.
You realize, just as a note to the to whoever is going to be compiling this, we're sort of flitting around between practice management and other sections. So just so that you're aware of that when you're transcribing, we will rearrange. So not to worry. But that's okay. That's okay. You know, I
I
think.
have a question for Mile.
Yeah.
Mile, that was you, right? Who just said that?
That's Chris
Yeah.
Sint.
Yeah, it is, Chris.
Okay. So going back to exactly what you said,
Yep.
you know, your staff spends a lot of time going through how peroxide works, why they shouldn't put it directly into their eye, how to use the system. Do you think in your practice you spend relatively more time having your staff explain peroxide to your patients and how to use it than they spend using a multi-purpose type solution? And do you think that probably plays into the compliance a little bit more? Because I too find that patients using a peroxide system tend to be a little more compliant to the system, but it may be because I've driven compliance home to them a little bit stronger.
Yeah, Chris, great question. So my staff members don't actually talk to them about the mechanism and how it works. They talk to them about the importance of doing what they're supposed to be doing so that everything gets neutralized, but they don't really go into some of the specifics.
The mechanism of the container.
Yeah. Most of it actually isn't that much longer than discussing multi-purpose solutions. The one reason why I would say yes, just a little bit, is because they'll physically take it out, and we have a clear care container, and even in the exam room, so that they actually are taking it out of the trial kit and showing it to the patient, which they don't necessarily do with the multi-purpose solutions when they're actually describing it to them. So it does take maybe just a little bit more time, but it's because we're going into the box and physically showing them the case and how it's different.
Good. That's a good point.
It may be more extensive, or
It probably is, just because it's a different system, so it requires a different container to actually take care of the contact lenses.
All right.
I think there's a mental image that people have of hydrogen peroxide. Many of them have used it to disinfect their countertops. They've used it for different things, like, you know, they get a wound and they put hydrogen peroxide on it, and so they know that it's powerful stuff. And I think if you warn them that if they get that in their eye, I mean, it's not going to kill them, but it may make them want to die. And the other,
do you tell that to your patients? You may want to die.
We actually tell them, we say, you know, if you get this in your eye, I said, there's a reason we use it. It kills bacteria very thoroughly, but if it gets into your eye, it will damage the cells on your eye exactly the same way. We're talking about the safety of the system. The other thing that someone mentioned is about topping off. I find that to be I mean, there's the only way you can tell if a patient is lying is if their lips are moving. But by and large, you can sort of tell if people. You have them bring their case and you have a condition. You ask them open-ended questions to find out if they are topping off. And we find that a lot of the people that develop infiltrates, in fact, that's their problem, is topping off. But I think that people are almost afraid to top off with peroxide. I mean, what would they put in there? They just add extra peroxide?
And yet it's surprising that people do top off with peroxide.
Whoa. Okay, enough of the sidebar conversations. Let's focus on maintaining some decorum here, ladies and gentlemen, okay?
Let's focus on art again.
Yeah, so now I will ask the questions and you guys will answer them, okay? So let's, I let you go because it was a really good conversation. I was enjoying it, but we really do actually have a job to do. So let's focus. Bill, you live in Canyon, Texas, and it's one of probably the highest allergen-laden places in the universe. Do you recommend peroxide because of its superiority in patients who are sensitive or allergic? I know that that's really been a mantra that I think most of us have expressed and believed. Do you find that to be true?
I do, and I find that people who tend to be to have allergies, to have atopy in general, are typically going to be people who may be more prone to develop a sensitivity to the components in solutions, in multipurpose solutions.
Well, I
have yet to find someone who's allergic to hydrogen peroxide.
Well, the question is, and I'll ask. Actually, I'll open it up to the panel here. Is that dogma, or is that reality? I mean, I haven't seen a sensitivity to an MPS preservative in a long, long time, but yet I also tend to think peroxide when I see an allergic patient. Chris?
Yeah, absolutely. And quite frankly, not just allergy, but many ocular surface diseases. Lid disease, allergy, and dry eye, I tend to shift patients more in the direction of peroxide, particularly Clear Care with its additives, and I find them to be quite comfortable. But I have not seen a clinical trial to support that. It's really been my anecdotal experience and my successes that keep reinforcing that practice.
Well, that's interesting. You said dry eye. What's your rationale for that?
What really, in my mind, brought Clear Care to a great place in practice was the transition to a one-step system, which improved compliance. But the addition of Pluronic 17 R4 as a surfactant,
right.
Really an excellent additive. It does a lot of interesting things that previously hydrogen peroxide systems did not really tackle in terms of aiding in the wettability of a lens through the actions of things like micelles, which are very analogous to macrophages, to clean up debris on a lens, to really go after lipid and cholesterols on lenses. Now we have the advent of silicone hydrogel lenses. Those things are problematic, and I think that additive has really assisted in dry eye practice with contact lens wearers.
Interesting. Anyone else have similar thoughts?
Well, I typically think of, well, let me back up here. The multi-purpose disinfecting solutions that we have at this point are so highly advanced, and they have these surfactants that are so advanced and are built for these difficult type of patients that I actually position Clear Care peroxide systems at a little different place than you, Dr. Levens, in that I'll usually go MPDS first, and if there's any what I see as a perceived hypersensitivity to the solution, or if there are any ocular allergy issues, I'll usually think of peroxide systems. When I see allergy patients, these patients too, it's not like their main symptom is itching. Their main symptom is the redness, but when we look at these contact lenses, they just physically look different because those eyes are more active, they're producing more mucus, they're producing more discharge. These things tend to deposit on contact lenses more readily. And I just find that for those patients, oftentimes it's easier to move them to a peroxide system and really attempts to keep them cleaner for the reasons that Chris was actually talking about. And for those patients, I have a high rate of success when I do switch them to a peroxide-based system.
You know, it's interesting. My experience is similar to yours as well, although I certainly understand your perspective. I find that for patients that tend to have dry eye, because of all the advances in the MPS, advanced MPS products like OptiFree Pure Moist, they're really optimized for patients that have dry eye, you know, just in terms of maintaining a wet surface. Peroxide does work well, you know, for many patients, but it tends to be not my first choice for those patients. But I think that's the beauty of having so many choices. I think that's, you know, that's a really great thing. Go ahead, Chris.
Art, I'll echo what you're saying here. I think a lot of it comes down to the surfactant that is used and the compatibility of the surfactant with both the contact lens and the ocular surface. And so the drier the patient, the more the surfactant in the contact lens solution is going to matter. And so typically if I have somebody who's very dry, I will put them into a product that has a highly effective surfactant, like you said, Pure Moist, first, and then see, you know, how they react to that before I necessarily change them to a hydrogen peroxide system. I think it really makes sure you need to match your solution to what the actual problem is. Everybody thinks that they're sensitive, and everybody thinks that they have allergy. But the fact of the matter is most of these people who are sensitive or have allergies actually have dry eye.
That's, you know, that's a great point, and I think my experience has been that every single patient that I've ever seen thinks that they have sensitive eyes. So it's just it's part of the human condition. I want to get into disinfection in a fairly big way because certainly that's a strong suit of peroxide. But I do want to touch on one thing, and that is peroxide has a very strong reputation for cleaning. Aside from the additives, which have helped that, the surfactants, does the panel see peroxide as a very, very effective cleaning solution? Mila, you're sort of nodding.
Absolutely, Art. It's actually one of the reasons and the logic behind me using it for those patients who may have seasonal sensitivities like seasonal allergies, those patients who have GPC and don't maybe have a daily disposable option to transition into. This solution is a phenomenal choice for these patients, and it is so because, again, we know the disinfection efficacy is high with hydrogen peroxide systems. It sets the standard or the bar that everything else is kind of compared to clinically. But in addition to that, we see it clinically. These lenses just come back and they're cleaner. So, Art, from a practical, clinical perspective, I just see what the data actually represents and shows us with patients.
That's a great point. You mentioned GPC, and it's not something, you know, we talk all that much about anymore, but I remember when GPC was rampant, peroxide was our go-to solution. And peroxide was very effective. You know, Bill, I know you're the king of creating GPC, and it's really GPC because it's Texas, so it's giant papillary conjunctivitis. What are your thoughts?
Well, my thoughts are that the lenses look good, but I've always wondered why. I mean, there's not really a surfactant in hydrogen peroxide. So my question is, does the hydrogen peroxide lyse the cells or the debris that are actually on the surface? Because you know, these patients are expected.
Peroxide penetrates lipids.
Yes.
It also breaks down the organic substrates and protein bonds. So it definitely removes, and I learned that in optometry school, that I still remember that today, that it breaks down, that's why it's good for GPC. Breaks down the protein bonds, denatures them, and removes them off the surface.
Right, which, exactly. And I think clinically, my experience is probably similar to everyone's. I mean, I've had really good results with that. And in the old days, Miraflow. You know, Miraflow and peroxide was really the way to go.
And that's where we started from, you know, with, we used a separate surfactant cleaner along with the peroxide. And so there was this habit that developed with peroxides of this rub and then, putting it through the system. And I think many doctors out there still use a rub, rinse, and soak system with peroxide, whereas patients may see one bottle solutions as pop a drop, take the lens out and just throw it in without doing that separate rubbing step. And so I think that, again, we very frequently will include a rub step in with the peroxide system.
Well, that goes back to the comments I think that Neil made, that I think that we tend to help patients comply because we tend to be more verbose in our directions with peroxide. So we actually do help with compliance. By the way, for those of you who are on the phone, we've just entered a romantic period. One of the lights went out, and it's gotten very moody and romantic here. So I just wanted you to know we're having a slightly different experience than you guys are. But I think that cleaning, compliance, patient issues all go hand in hand. One of the things that Loretta mentioned, which I think is critically important, is that microorganisms want to survive at all costs. So they very rapidly develop strategies for survival, and there are some organisms that produce enough antioxidants, catalase, peroxidase, to survive the initial burst of peroxide in a catalytic disc system, and then they survive. And some of them produce biofilms, and you're one of the world's experts on those issues. So what are your thoughts on peroxide relative to biofilms? I know you've done research on it.
Right, well, as Mila said, really peroxide seems to be the gold standard, and the MPSs, MPDSs seem to, some function just as well, but that is the gold bar that we're trying to achieve. So we've done work both in fungal and bacterial biofilms using clinical isolates. So, came from contact lens inflammatory conditions or contact lens ulcers, microbial keratitis. And we found that all of those strains that are clinical isolates form biofilm. By the way, that's not true for some of the FDA strains. The fungal isolate that's required in the FDA for Fusarium solani doesn't even form biofilm. It's a very old isolate, so it's kind of an irrelevant organism anyway. So all of the clinical isolates form biofilms on contact lenses. That was our first step to show that indeed they form on contact lenses. We haven't done work to show that they form inside cases, but every study you can read back to the 70s talks about biofilm in contact lens cases. So we developed a model to look at disinfection efficacy on biofilms formed on contact lenses, and different contact lenses, not just the leading silicone hydrogels, but some low decay lenses as well. And for bacteria, peroxide, and the one we tested was ClearCare, was the only one that was able to eradicate biofilms formed by Pseudomonas, Serratia, and Staph aureus. That would be Serratia marcescens, Pseudomonas aeruginosa, for the record. Most of the PHMB preserved systems, actually all of the PHMB preserved systems, could not even touch the biofilm. And some of the OptiFree products that we tested were efficacious against 2 out of those 3 organisms. So, but peroxide was the only one effective against all 3. And that is different than the planktonic experiments where you're doing it on free-floating organisms, which is what the FDA requires. So what we know about disinfection when it comes to planktonics is that they all do a great job. But you start to see differentiation when you look at the resistant biofilms that definitely form in cases, and these organisms encapsulate themselves and protect themselves from the environment, make it much harder to disinfect with both the host immune responses as well as from disinfection products that are on the market. For fungal biofilms, we also found that only peroxide, and we looked at Fusarium oxysporum and Fusarium solani, and only peroxide was effective against both of those isolates on all contact lenses tested.
That's the work was phenomenal. By the way, it was really groundbreaking work, and you drew attention to something that is I think a very, very serious issue, and that is our disinfection standards are based on planktonic bacteria, and the reality is that we deal with biofilm forming organisms more often than not. And you're exactly right. There are studies that go back to the 70s that show massive concentrations after time, even in peroxide, of biofilms. So I think one of the things that that I just want to discuss very, very briefly is the need for a gold standard, from a clinical point of view, because we can't rely on FDA standards at this point, and we have to take control of that from a risk management point of view in our own offices. And we need to be looking for gold standards, whether we're looking at peroxide when it's appropriate for a patient or we're looking at an MPDS. We need to ensure that those products function adequately and optimally. So any thoughts on how you approach that? And I'll open this up to to the phone group as well. Do you take that seriously? I certainly do. I don't see FDA standards as having as much relevance as I once did, and so I'm looking for products that I consider to be gold standards, and certainly ClearCare is among them.
Well, I guess I'm just.
Okay, Bill.
Go ahead, Bill.
Go ahead, Bill, and then me.
Okay, so when we actually sit down and there's been numerous studies that show that when we compare the FDA testing to real life, it's almost like a fairy tale because you're dealing with there's no lens in the testing, like in the vial or the test tube. We know that the lens itself can make a tremendous difference, maybe by absorbing bacteria or whatever. So we know that for the fact. We also know that these are basically organisms that have been used over and over. Whereas, I had looked at an article earlier where they actually took this strain of amoeba that was taken out of the tap water of Chicago, and it was apparently a terrible, very, very difficult to kill organism. So I think that we can. I won't say that it's irrelevant, that the FDA testing is irrelevant, but I think that it should probably be reevaluated for something that's more purposeful and more impactful on the day-to-day practitioner and our patients.
Well, I think that that's underway, but it's taking a long time. Eventually, we will have better standards that are more reflective of what we're seeing in the environment. But I think it's important for our colleagues to recognize that as a potentially big issue. One of the other things that I did want to talk about is different peroxide choices that we have. Once you've made a decision to use a peroxide system, are all the peroxide systems the same? We have generic systems now, and certainly I think there are differences. Has anyone found significant differences? Are patients using generics?
I can tell you practically, we've had more of a few bizarre situations with the store brand products in terms of what I'm going to describe as the quality of the caging or the systems that they're actually contained in. I've actually had a few patients come back and say that they used the store brand products and they broke on them. So they just decided to continue with the ClearCare because in their minds they felt that it was just structurally put together better, and there was a certain level of trust in that. Certainly from a historical perspective, and again, I'm speaking from 10 years, not however long you've been practicing.
Not 100 years.
But we've had such good, strong success, and we've built up this strong level of trust with the ClearCare brand, knowing that we're going to get a certain level of consistency every single time, which is strong disinfection and very clean lenses. Very clean lenses.
One of my really big concerns, and it goes back to an article I wrote in 1990, is residual peroxide. And one of my concerns is that some of the products have much higher levels of residual peroxide, and I think that can be an issue. Chris, Sint, any thoughts?
Well, keep in mind our body has a way of dealing with residual peroxide. Okay? Some people are going to deal with it better than other people. Some dry eye situations may not deal with it as well as people who have a very thick and robust tear film. And so the amount of residual peroxide that's left in the container is really going to depend on the eye that you're putting it on. So obviously, if we can make the residual peroxide as low as possible, we're going to be able to have comfortable and happy eyes on the greatest number of our patients out there.
Well, I think that's a great point, and the point of this series of case studies that I wrote up was really looking at relatively low levels of residual peroxide producing keratitis in some patients, and actually one patient went on to have a bacterial ulcer on a cruise ship, of all places. So the concern is we want to achieve 0% residual peroxide ideally. We can't always do that, but the less we have, I think, the better it is for the patient. So I'm concerned about generics. I'm concerned about strategies that would increase residual levels, and I think we all need to recognize that can be one major issue when it comes to peroxides. Yes.
I can't say that I know much, this is Loretta, about the peroxides that are generics on the market. I was actually pretty surprised once when I walked into Target and saw a bunch of, peroxide systems there. So I think the reason I'm not concerned about it is because my patients never tell me they're using it. Going back to the compliance issue of peroxide users being compliant, it's actually the only product that they can name by name. When they come back into the office, and we always ask them, what solutions are you using? The technicians always, one of the first three questions they ask. ClearCare is always named by name, whereas the MPSs, they can't remember the name half the time. So I don't think any of my patients, I shouldn't say any, but most of my patients do not buy the generic product.
I had a colleague call me recently in the last couple of weeks, and it's funny, generic peroxides can do funny things to your lenses, and including turn the lenses pink.
Yeah, nice pink. Yes.
A nice pink color, and I think maybe we've all seen that, happen. So, the purity of the peroxide actually matters and actually is important.
That is true. I have seen a number of those cases and especially when patients use over-the-counter store-bought peroxide that's for disinfection. You can add all kinds of weird things. So I think the key here is just because it's peroxide, just because it's ClearCare, we don't have that cushion anymore where we can assume that they're only using ClearCare. So we have to reinforce specifically the product that we're prescribing, and just make sure they understand that we don't want them using a generic. Chris,
I just thought Loretta's comment was really interesting. And I find the exact same thing. Even though I practice in a very poor inner city patient population, patients are very brand loyal with ClearCare. I also don't have a patient respond, what type of cleaning system are you on, say hydrogen peroxide. They specifically call out ClearCare. And so I would be very much willing to say that I don't believe my patients are using generic peroxide.
Right. And the other side of that coin, and in ensuring that they're not, even though we're believing that they're not, and you're probably right, many patients do make that distinction, is also instructing them to replace cases and not to top up. Just touching back on the compliance issue, even though we think, why would a patient top up with peroxide? They do top up with peroxide, and we know that that's a strategy for massive failure, which we have to certainly be cognizant of.
Can I just add one thing
You certainly can.
what everybody was saying? I think it's important. I think one of the ways that we try and safeguard against these types of things in our practice, and although this sounds kind of basic and rudimentary, it is so effective, and it gives us a true real picture of what our patients are doing with their contact lens care. Our message is simple. Whenever somebody calls our office to schedule an appointment and their contact lens wearer, they're told to bring in their contact lens case, their solution, and any products or drops they might be using to care for their lenses or their eyes. And then when we call them the day before their appointment, we give them that exact same message. And the reason why is we just want to safeguard and make sure that people are actually doing and using the products that we ask them to use and how to actually use them.
Actually, that's a very, very good strategy. I think it varies from practice to practice. In some cases, you wouldn't see that at all. Patients just wouldn't comply with bringing in their cases and their lens care products. It's just the nature of that. Okay, so let's see. One of the things I wanted to talk about.
You had enough questions on your thing.
No,
we're 2 days there, aren't
we're taking a momentary break. I have a lot of.
Are you going to talk about
oh yeah,
the
okay,
risk
I
of
got that.
amoeba? I mean, how big a risk really is it?
Say that again, Bill. Sorry.
Are you going to talk about the real versus the perceived risk of amoeba?
Actually, I will be talking about disinfection in more detail again. We're going to go back to it in a sec. I just wanted to comment on, has anybody seen Safflon as a product? They sort of made massive inroads. I know in the private label market, they actually sell their own products, but they literally are on the market for peroxide with a number of different brands. Has anybody encountered any of those products or aware of that?
I'm aware of it, Art, but I haven't had any patients utilizing those products.
Well, I think you actually have, because they market it under other brand names. So, I think that's an issue. And the product is, not anywhere near ClearCare in terms of its quality, at least in my experience. But it's, and that's interesting, because we often don't know what's in the bottle. And that's, to me, very, very frustrating. As a clinician, I want to know what I'm prescribing. And I think in this time of generics, I think that becomes increasingly difficult. I wanted to talk about, before we get back into making choices and disinfection as an important element, I wanted to talk about biocompatibility. One of the big issues that we've had and we faced for probably the past 3-4 years is corneal staining, and I know corneal staining has been minimized because of commercial interest, and the significance and relevance of it has been questioned. To me, corneal staining is a big issue. What have been your experiences with peroxide and corneal staining? Bill? Chris?
Go ahead. Chris, go ahead.
You can get corneal staining in any contact lens related corneal staining in any patient who wears contact lenses. Okay, so, there's always going to be some level of corneal staining if the patient's actually wearing the lenses. Certainly, we know that certain brands of contact lenses and certain brands of solution are going to induce more corneal or conjunctival staining. And in the research that I'm doing looking at confocal microscopy, we definitely see relationships between the level of corneal staining and the amount of dendritic cell infiltration into the tissue. So we know that corneal staining, based on the work that I'm looking at here, that on some level corneal staining is driving, at least driving inflammatory cells on a subclinical level, if not on a clinical level itself. So, going back to your question, do we see corneal staining in patients that use ClearCare? You absolutely can see corneal staining in patients who use ClearCare, but it may be lens dependent, but it may not be to a level that we would consider clinically relevant.
Yeah, well, actually, you mentioned dendritic cells. I just want to reinforce for our readers that dendritic cells are inflammatory cells that appear within the cornea, and maybe
antigen-presenting cells.
Yeah, okay, and I want you to go into it for just. I try to simplify it, but I want you to go into it for just maybe 30 seconds. Just describe your work briefly, because I know it's not relevant specifically to peroxide, but it's more general in terms of contact lens disinfection systems. But it is of interest.
So the research that I'm doing, we're looking at various contact lens solutions and various types of contact lenses, and to see how corneas respond to wearing contact lenses. So we've looked at established contact lens wearers, and we've looked at neophyte contact lens wearers to see how they respond to wearing contact lenses, and to see if there's a relationship between contact lenses themselves or contact lenses and specific solutions.
Okay. Well, you made comments about staining. I find that peroxide produces probably the lowest levels of staining of any of the solutions that I use. Some produce very low levels to start with. Some produce very high levels. Peroxide, Chris?
I totally agree. I would say absolutely is one of the least in terms of across brands, across lenses, across lens materials. I've made it a habit in the past probably 18 to 24 months after doing some clinical trials, specifically looking at corneal and conjunctival staining, to really look at the raw cornea before I put anything in, before I put in fluorescein, before I put in any type of a drop. If there's significant keratitis on the or staining on the conjunctiva, oftentimes it's very, very visible before anything goes in, and the fluorescein oftentimes confirms what I've seen on the naked eye. And it's concerning to me as a practitioner, but I would absolutely agree that I see consistently across brands low levels of staining with hydrogen peroxide systems in general.
What you just said was absolutely, I think, critically important. We sometimes forget that fluorescein is just a disclosing agent, and what you're seeing with fluorescein is just an exaggerated version of what you should be seeing with naked light if you're a good examiner and take the time to examine. So when we talk about staining, we really are talking about disruption of the cornea. I'm not talking about staining, as an artifact. I'm talking about staining as something that you can see without fluorescein, and I think we need to make that distinction. Chris?
I want to really, if I could go back to looking at using the confocal microscope and looking at staining and inflammatory responses on a cellular level, I really do want to drive home the fact that peroxide is not necessarily better than certain MPS solutions. Okay? MPS solutions can be very effective and can be very safe with staining and with dendritic cell migration into the cornea, just as peroxides can as well. They're not necessarily better than certain MPS solutions. MPS solutions can be very safe. However, there are certain MPS contact lens solutions that do cause staining and do drive dendritic cell migration into the cornea and into the conjunctiva. Does that make sense? On a cellular level, it's not necessarily better. It's very good, but there are MPS solutions out there that are very good as well.
And I think that's an important point. And I think for the people who are going to be reading this, we should make it very clear that what you're talking about is polyquat aldox producing relatively low levels of staining, very similar to peroxide. And you're talking about PHMB-based solutions producing significantly higher levels of staining. And that staining isn't just staining by itself, but staining is also inflammation. And staining is also changes in comfort.
Staining is definitely linked to inflammation.
Right,
It's also
yes, Bill?
definitely. There's an article that we read in our homework assignment, and we knew this sort of inherently, but now there's evidence that shows that staining is definitely linked to discomfort.
Correct.
We've all seen people who come in that have tremendous staining and they don't really have discomfort, but by and large, there is an association.
And I think that may be one of the reasons why I find that peroxide is so comfortable for patients. And I think that's traditionally been well accepted. Neil? One
thing about corneal staining. I've been looking in a large trial I just completed at corneal staining very closely, and in peroxide users were one particular arm. And staining can happen in a peroxide user, but it's different. You're very right, it's very, very low percentage, very, very low incidence. But when it does happen, it doesn't look like solution-induced corneal staining. It looks mechanical to me. They're larger macropunctate stains in the inferior third of the cornea or central cornea. It's very lens-related, very mechanical. And in those patients, guess what they do better in? an MPS that has, some of the wetting agents in it. So it's similar to the dry eye discussion you were having before. So it can happen, but I think it's not solution related at all. It's mechanical, perhaps dry eye.
Interesting. You're going to comment then?
Well, I was, but I think I'm just echoing what everybody else is saying. I do find a high level of efficacy with Clear Care, and I do find low levels of corneal staining. I mean, it's just something that will reliably do what it's supposed to do, period. Loretta, I would echo what you're saying. Sometimes you see some mechanical issues, but those look very, very different clinically than that solution-induced staining because of where and how you're seeing it on the cornea. Fortunately, we have this option for those people who do have hypersensitivities. Fortunately, we're fortunate that we have this option for patients who do experience these issues, who do have all of these other issues that we talked about, and we can bring these patients back into a system like Clear Care that just will fix a lot of these, I'm not going to call them benign, but these almost problematic issues that will at times cause patients to find contact lenses more burdensome than beneficial, and we can just bring them back to a level of normalcy. I'm wearing my contact lenses again and I'm not aware of them anymore. And a lot of that has to do with almost everything that we've been talking about. Corneal staining, conjunctival staining, clean lenses, and a high level of disinfection.
Well, one of the things that we're actually not talking about that we probably should, and to your point about staining being very different with peroxide than it is with MPSs, is we're not seeing uptake and release as an issue with peroxide. When we look at products like PHMB based disinfectants, we're literally seeing uptake into the lens and then release back out into the precorneal tear film, which reaches toxic levels and produces frank staining, and at the same time reduces potentially the efficacy of the disinfectant through uptake. Yes.
I'd be careful. I'd be careful about that statement going into print.
Oh. When have I ever been careful about statements going into print?
The reason is because there's some evidence that there is uptake and release of the peroxide. It's just handled differently on the eye, and different materials are going to handle it differently. The thing is that our bodies have ways of handling peroxide that's released back into the tear film that is very different than the way our body handle say PHMB as it's released back into the tear film. And so our bodies don't have a way of neutralizing the other disinfectants necessarily, whereas it does have a way of neutralizing the peroxide. And so, more evidence, there needs to be a lot more studies out there, and in a year or so we'll be talking things a little bit differently. But I would say that peroxide uptake and release is more of an intelligent biocentric design than say some of the other chemical disinfectants.
Well, I would agree with one caveat. If you have a system that neutralizes peroxide down to 50 parts per million, the equilibrium that will be reached will be that there will be 50 parts per million in the lens, more or less, and that is so relatively little that I think, to your point, the body.
Unless the plastic is hiding it.
Yeah, well, that would assume, and then we don't know that. That would assume that some.
And that's where there's going to be we'll know more.
I sense research. I wanted to, we still have some time, but one of the things that I wanted to talk about was deposits on lenses, and that's an area that I'm actually very much involved in and planning some research. And does peroxide reduce deposits, and what's the clinical significance of that? What?
Depending on the brand, as we talked about earlier, the surfactant additive to Clear Care does have an impact on deposits. It is dependent on lens material. It is slightly more effective on certain lens types than others. The whole class of silicone hydrogels is still very, very challenging. So it has an impact. I'm not sure how substantial it is, and it does vary across brands of lenses.
Yeah, that's interesting. One of the things that I'll share with you, and I don't know if this will make it into print, but the less the deposition we get on lenses, particularly silicone hydrogel of lipids, the more stable the tear film. So when you have a product like a peroxide that lyses lipids and doesn't allow the surface to become increasingly hydrophobic, more lipids stay in the tear film, and lipids actually are quite functional within the tear film. So I think that's an area that we're going to be looking at in some detail.
Were you saying, Art, that there are certain deposits that can create the lens surface to be more hydrophilic?
Correct, and silicone is innately hydrophilic. Some of the different lens materials have a lot of silicone exposed to the lens surface, which produces an attractant to lipids, which are hydrophobic. And the more hydrophobic the surface becomes, the more lipid is attracted to the surface, and it builds up and it builds up and it builds up. And if you're taking a lens that's used for a month, and you're taking a lens that's already biased towards hydrophobicity, you're actually just spiraling things further out of control. And it's an area obviously that we're quite fascinated with. So peroxide is a strategy, I think, for minimizing at least some of that hydrophobic buildup, because its ability to break down lipid deposits. But that's an area for further research. We've talked about peroxide and peroxide. Most of us, I think, when we think peroxide, think clear care, because to me it is the gold standard. It's what I think of. I use the term peroxide, but I'm really thinking of clear care. The question is it different than just peroxide, and what makes it different than peroxide? Any thoughts? Any takers on that one?
I've actually asked the people in industry that question, and they tell me there is a tremendous difference based on, I guess you call them complete, not pollutants, but there are molecules in, say, store-bought hydrogen peroxide that are not good for the contact lenses, that cause these color changes, etc. And those are definitely not present in clear care. They've gone to great lengths to make sure that what you're getting is hydrogen peroxide, and just about that's all there is.
Well, what about
optical grade peroxide is very different than commercial grade peroxide.
But let's get more specific. What about relative to some of the generics I mentioned, or to some other products? We think of peroxide as peroxide. Is clear care a unique formulation?
As we know, when we think about other generic medications and drugs that are on the market, even though aspirin may be aspirin, right? It's the other things that are added to the product that will change how that product works. So it might not even be what's considered an active ingredient. It may be some of the quote-unquote inactive ingredients in there. And this is true whether you're talking peroxide or whether you're talking multi-purpose solutions, right? So the biggest thing that makes a difference is the addition of, say, surfactants or the floronic into the product.
Good. Okay.
One of the other things that we didn't mention, but that we probably should talk about, is residual peroxide levels, and that they do vary with the age of the disc.
We actually did refer to it a little bit while you were sleeping, Grant. While you were sleeping, Grant. We did talk about it. And again, I think that's very, very important. I think that's going to become a big issue as peroxide becomes more common. And again, I guess most of you realize that peroxide's doubled since the fusarium and acanthamoeba outbreaks. I don't know. It's important for our readers to recognize that peroxide has actually doubled in use. From about 8% to about 16%. Largely because of that trust in the product. And but I think that the point that Chris, I think you made very, very nicely, is that there are significant formulation differences. The clear care wasn't something that was cobbled together by accident. It was actually made very, very carefully. It was formulated to be an advanced product, which I think has made a difference. So I want to talk about corneal infiltrates. That's a very, very hot topic. I know it's one that's near and dear to your heart, Loretta, and it's certainly, I think, near and dear to many of our hearts because of the barrage that we've been seeing about corneal infiltrates. And then I want to end this by talking about practice and patient management. But are corneal infiltrates that big an issue, and where are corneal infiltrates coming from? Is it a solution-related thing? Are we seeing it with peroxide? Is this, a sudden epidemic? And what's going on, Loretta? It's by the way, that's sort of a hand grenade. Loretta?
Well, infiltrates are a very big deal in extended wear. In extended wear, the rates are really not acceptable, I think, for the average practitioner to be able to handle and manage that. In daily wear, I think the rates are rather low, very low, and manageable. So we don't see as many symptomatic infiltrates. We see asymptomatic infiltrates in daily wear at a reasonably low level. There are these hypersensitive patients that seem to be sensitive to lots of things, and peroxide's probably a good option for them, or daily disposables. Across the board, in large-scale retrospective studies, peroxide is always has lower infiltrative responses than any MPSs as a group. So Chalmers has done at least 2, if not 3, large retrospective studies of thousands of wears, and it's always been significant. Peroxide has definitely a lower rate, as well as the studies that are coming out from Australia. The current studies also show that peroxide has the lowest rate. Now, it's not immune to infiltrates. So infiltrates can also occur in peroxide users, but when they do, they're not due to hypersensitivity to the product. They're due to patient factors, and that is usually bacteria that come from the patient, either their hands or their lids. And we've actually even been able to isolate it to normal flora. So there are patient factors we can't get away from in infiltrates. And regardless of the product that they're using, because the last thing that touches the lens before it goes in the eye is the hands or the lid. And if there's significant bacteria on the hands or the lid that cannot be handled by the system's immune, well, actually it is being handled by the host's immune system, and what you're seeing is the infiltrate coming in and attacking the bacteria on the lens that came from, the patient's own handling or their own lids. So you can't get away from infiltrates because of patient factors.
I think that's a great point. I think we've hyper-focused on contact lens solutions as being the source of many of the infiltrates that people are reporting, which is odd because you have practices where there are no infiltrates at all, despite using a solution that's been questioned, and yet other practices seem to have a lot of them. And in my experience, the vast majority of infiltrates are from staph exotoxin or from normal flora, and you're exactly right. It's the body doing what it's supposed to do. It's responding to those alien proteins by sending in white cells to defend against those things. And to your point, peroxide produces the least amount, probably, at least in part because of handling issues and things like that. So I think it's multifactorial, and there are many, many factors. But again, it goes back to the gold standard. If you have a patient where you have a history of infiltrates and you're concerned, certainly peroxide has the lowest rate of infiltrates.
And I think what Loretta just said
It's
is
Chris.
important. I am more likely to prescribe a peroxide-based system for my patients that have what I consider to be significant lid disease, because I think they will have the best reaction to that, as opposed to, say, the dry eye patient who may actually do better in an MPS solution. And I think that goes back to, it's important to not have sort of this knee-jerk prescribing. And one thing that really bothers me when I see patients or doctors talking about peroxide is they either will frequently use the word "it's my problem solver," meaning that they only use it when they see problems and then change patients into peroxide, or they have this knee-jerk reaction and prescribe everything.
Yeah.
And I think we need to focus on intelligent prescribing. which is focus on the patients that may most likely have the problem, put them into the first line choice.
Okay,
We're getting an echo on you,
we get-
Chris.
Chris, have you suddenly moved to Beijing? All right.
I am giving a lot of feedback.
Okay.
better.
Well, so no, it's better now. Everybody separate their phones. Bill, keep the dog quiet. Oh no, actually, it's me feeding back.
You want me to be quiet or you want me to say something?
All right, okay, everyone on the phone, guys. Shh. Chris.
Yeah, I think some folks are barking up the wrong tree a little bit, looking at solution specifically causing infiltrates. I was asked that question several times the past couple of years.
I'm pretty sure that speakerphone is on the other side. That's what we're hearing.
Yeah, if someone has a speakerphone on, could you turn it off? Bill.
I don't have a speakerphone.
Okay, Chris.
No.
Okay. Well, we've troubleshot that.
My phone. I don't know. I suddenly can't hear you. What is that?
Okay, that's very strange. Okay, 319 is Chris. Okay. Okay, you can't hear anything. Echo. Okay. Hold on. Okay.
You're the echo. We're the echoes.
We're the echo. So how do we? Okay, how do we deal with? So anyway, so as I was saying, we were walking down a long road. Anyway.
Yeah. What I was saying is I think that we're barking up the wrong tree a little bit looking at solution-induced infiltrative response. I was asked that question several times over the past couple of years, and I hadn't seen it personally, so I dove into our EHR and pulled every time an infiltrate was diagnosed and tried to look at the history, and I didn't identify it being related to a solution. The lion's share of our patients use the Opti-Free Pure Moist brand with a smattering across brands after that, and I have absolutely not seen that. And in fact, just exactly what Loretta and Chris said, I believe it absolutely to be lid-induced predominantly.
And it's frustrating to me personally because it's become a commercial issue, and it shouldn't be. It's a health issue. I want to segue over to what?
It's still echoing.
Yeah. Guys, if we can't fix it, we're going to have
Maybe we
to.
can end the call and restart the call.
Yeah, maybe we should. Yeah, why don't we end it and restart it? Okay, we're going to break.
Hey, if all else fails, I mean, we got approximately 15 minutes left. And if that's too much trouble, we can finish here and they're done. Yeah.
Yeah.
You get exactly.
You guys still taping?
No, we just got.
Oh, okay. Because I just want to really focus on practice management for 10 minutes.
Well, we can get comments from them.
After,
Email.
by email afterwards, yeah. I think that would make the most sense. Besides, they were disruptive anyway. Okay. Wish them well.
You know what? I don't know if we should go here, but my data show that it's not the blepharitis patients that have the staph hypersensitivity. You can't actually see the staph, so it's kind of you don't know how to
What it is.
attack it, but you don't know how to attack it against identify your patients. Or how to
Yeah, it's interesting. It's sort of counterintuitive because you would think who has the highest population of flora or overpopulation of flora would be meibomian gland dysfunction.
But I think that those patients have found a way, their bodies have found a way to deal with it, and blepharitis is the way that they found the way to adapt to it, so that the lid has evolved over a way, and you see the blepharitis, which could be the anti-blepharitis response to the staph, but that's kind of the way it's contained it, or it's already developed its own immune response. It's really bizarre. All those patients that to me look hypersensitive with all these CLPU scars everywhere, they actually have very clean lids. Amazing. Look next time. Next time you see a hypersensitive patient, look at their lids. They don't have horrible blepharitis.
So,
they
it's
have
fun-
super clean lids.
It's funny. I always assumed it was that they still had meibomian gland dysfunction. In fact, when Korb came up with his no MGD, I went, It's sort of now all of a sudden that made sense. They always had greasy-looking lids, but not really overt blepharitis or overt. Yeah, exactly. So that was exactly.
You almost, my point is you almost don't know until after the fact. So you treat everybody the same, and then you find these hypersensitive people, and then, they just do.
But to your point is even normal-looking patients have, infiltrates that are associated with bacterial.
lids.
Yeah.
That's the point.
So that's an even better point to me. To me, the commercial stuff annoys the hell out of me. We back on? Oh, good. Okay, good. So it does annoy the heck out of me. So one of the things that, and by the way, we're going to be changing some of the peroxide verbiage to clear care because one of the things that I realize we're doing is that we're talking about peroxide when we should be talking about clear care because we just said how we recognize that as clear care. Mila, you're really great at practice management. I know that's one of the areas that you're highly focused in, just in terms of analyzing what you're doing to be successful. Do you find that recommending a specific product, specifically Clear Care, has made a difference? Do you use it to differentiate yourself? Yeah. And how do you do that?
That's a great question, Art. And this is something that is critically important. In the era of commoditization, sometimes really the only thing that differentiates us from anybody else that practices around us is the way that we position ourselves and the perception of a patient's experience leaving our practice. Certainly we all put a lot of work into educating ourselves and understanding the best processes, but if we don't consciously put forth efforts to tell patients that, they're not going to know the difference. And one of the ways we do that is we take contact lenses very seriously. And we do very specific things to attempt to differentiate the experience. One of the first things I talked about was having people bring in their stuff. Although it seems somewhat logical, it's surprising to me how before we did that, how everybody thought they were using the solution that was actually on our countertops, and then when we actually made them responsible, we realized that they were using things that were significantly different. What I've realized is that people will not care for things more than we care for them. So if we take an active approach in understanding contact lens care and how it can affect patient outcomes, and we take the time to explain that to patients, which isn't a long conversation, but it's just something to let them know that we understand that this is what the best product or solution is for their specific eyes, and letting them know why it is and helping them understand how to properly utilize it. We've already taken steps to now decommoditize ourselves. So Art, in my mind, taking a generic grab bag approach and saying, well, here's some stuff that you can try and you can use which one's best, is not the best approach. In my mind, the best approach is clearly defining what you want that patient to use. And if we're deciding that peroxide is in that patient's best interest, we're making it very clear that we want them to use Clear Care, and we're educating them on exactly the way to do it. So in my mind, it's a way to decommoditize the services that we're providing to those patients.
That's a great point. I think I should also add that you two have the same issues in your clinical populations in terms of patient control and compliance. Do you find that it makes a difference in terms of that?
No doubt. And in fact, even though we're a big inner city clinic, we still treat our patient base as if it were a very large private practice with independent practitioners. And the same holds true in the discussion that we're having about contact lens solutions, holds true throughout the exam. What Mila said about building the doctor-patient relationship as strong as possible oftentimes lies in defining the question why. Why am I prescribing you this particular lens in your spectacles? Why am I prescribing this particular specific glaucoma medication for your glaucoma? Why am I giving you this specific contact lens prescription and this specific solution? It's all about why. And when those whys are delivered appropriately, the doctor-patient relationship really blossoms and the patient becomes more compliant, the practice builds, and everybody wins.
That's literally like sage advice. It's the one thing we still have left, in a crazy time where We have lots of issues that face us in terms of patient relationships and regulatory challenges. We still have the doctor-patient relationship if we choose to maximize it. And making sure that you take the time to explain why is all the difference in the world. And using products that differentiate yourselves and distinguish yourself the same way you want to be a brand, you want to use a brand, I think that's very, very important. When would you choose a peroxide system? I know we talked about that, but just in very short terms, give me like 2 or 3 patients for whom you would definitely choose peroxide. Let's go around the table.
Okay, so I'll start. 2 patients. One, somebody who I've tried on multiple or even one multi-purpose disinfecting solution where I suspect some type of sensitivity. Somebody who's coming in with excessive deposits and a known hypersensitivity to something in the environment, i.e. that allergy patient who comes in every May and September with problems or issues. Those are really 2 key patient demographics for myself, Art.
Perfect. Loretta?
Same thing, anyone that has shown any hypersensitivity to any other solutions. I also am a big user in RGP wearers and piggyback patients. I do a lot of piggybacks in mine. I do a lot of therapeutic lens fits, and peroxide, clear care in particular, being approved for both RGP and soft has been a great problem solver. Actually, that's my go-to product for ease of care and compatibility of the 2 lenses together. I find when I use an MPS and a traditional GP cleaner, there is actually a whole host of non-compatibility there. So I use it for them as well. And then anyone that I'm worried about from a hygienic standpoint, again, going to that gold standard disinfection efficacy, or if they've had an ulcer in the past, or anyone that I'm just for some reason clinically concerned about high levels of contamination.
Good point. Chris?
Once I have a patient that I believe is in a daily wear lens that they're using every single day, in all likelihood. And then I also have, and they've been wearing a multi-purpose disinfecting solution and still have some type of ocular surface effects that are symptomatic, I start to steer them to clear care. And I find it to be very successful.
Okay. When would you definitely not prescribe peroxide? When would you prescribe an MPDS like OptiFree Pure Moist?
Art, actually, OptiFree Pure Moist is my first line for patients. So I guess a better or more appropriate question for me would be, when do you prescribe clear care for your contact lens wearers? So it's OptiFree Pure Moist, usually first line for everybody, and then if there are any type of issues, then we resort to clear care.
Excellent.
The occasional wear.
Perfect. Exactly.
Occasional wear.
Occasional wear. And the reason why is buildup of potentially pathogenic bacteria in the biofilm on the case and lack of disinfection efficacy because of neutralization and having a lens soak in that. So that's a good point.
Also, I do have one more patient that I don't prescribe it for. People that travel a lot. Actually, it doesn't travel very well, I have to admit, in when they have their lenses in their case and it's not sitting upright. So we have come up with different mechanisms here, put it in this MPS for the interim, but that's just another patient population that I...
That's a great point. Do any of you use peroxide intermittently or clear care intermittently, like during seasonal allergy periods, and then go back to OptiFree Pure Moist, or do you stick with what you prescribe? Do you find it confusing to go back and forth?
I personally think it's a little bit confusing from a patient perspective. There are some patients that I have on that regimen, but most, if they are switching to peroxide, will likely stay with peroxide.
Okay. Yeah, and I see everybody nodding. One last thing, and then I have a surprise for you. It's a special surprise. If you were talking directly to your colleague, how would you empower them to change their practice, to deal with the coming issues that we're facing, specifically in terms of using OptiFree Clear Care or products that are discernible as different to the patient? Do you have any specific advice to how they could become centers of excellence in their practices?
Yeah, Art, I think whenever we're talking about, I liked how you put that center of excellence, and focusing on contact lenses and contact lens care, I think we just simply need to take the topic very seriously in our practices, because if we don't, our patients certainly won't. So the more seriously we take the topics, the more seriously our patients will care for those topics. Again, reiterating the whole, bringing everything into the exam with them is so critical, because this actually gives you the ability to visually say, yes, this is right, no, this is wrong, and this is how we need to change it.
Great. Loretta, you to me are the gold standard of science. You literally push the envelope. Can you translate that for our colleagues, to empower them in their practices? What you've done with your career?
Well, I'm a clinician too. I still see patients twice a week, so I'm in there in the trenches just like the rest of you. No, I think you mean how I would change.
Yeah, in other words, what advice would you give to someone who's just getting out of school now and starting a practice and
I see. Well, it would be to prescribe contact lenses and the products just like you prescribe medicines. Don't let them leave with a visual. Most importantly, a sample. If you don't have a sample, some other visual. I think it was Chris that says she literally has some kind of a prescription pad that she tears off at the end of their exam and says, this is the solution system that you need to be using. So I would empower my colleagues to prescribe the products that they feel are appropriate for their patients, just like they would prescribe medicines.
I think one of the most important things to do is embrace lifelong learning and constantly question yourself, question others, question what you see, question what you read, and that strengthens you as a practitioner and delivers the best care long term. Just as we talked about, as patients want to hear the why they're doing things and why you're prescribing, the practitioner needs to fully understand why they're doing what they're doing and not just as a matter of habit. Matter of habit and dogma is the easy way to go, but the best practitioners are constantly questioning what they do and make sure they're on the forefront, and lifelong learning would certainly push them in the right direction.
Yeah, I think those are great points. I think one of the things that I think to me is critically important is communication. And whenever I speak, I always tell the audience that my hope is that you don't just learn something from this, but you take home something that you can use to communicate to your patients, because communication is the essence of empowerment and really bonds people to you, which I think is very, very important. I hope that this roundtable has given you insight, and that insight will allow you to better communicate choices to patients and why you're making those choices and working with them to ensure their safety and well-being. Now, the homework assignment I have for you, and it's a relatively minor one, but I think it's very important, is I want you guys, and obviously you were chosen for special reasons, is to think back, and I sort of alluded to this here, to your most unusual case where ClearCare saved the day, or ClearCare made a difference, or ClearCare changed a life, or ClearCare saved an eye. And just write up a short little thing for a sidebar. Okay. And what we're going to do is focus on those little clinical pearls. And believe me, I know you're a clinician. I know you well enough to know you're a really good clinician too. So let's share a little bit of clinical expertise and let's kick some butt. Things are changing in our community, and I think if we all don't step up and try to direct things into an appropriate way for our colleagues and our clinicians, we're not fulfilling our destiny. So, good. Well, any questions that you guys have? Oh yeah, I do have one question. ClearCare has been sort of the red-headed stepchild, and SEBA in its lifetime really never promoted it very aggressively to consumers. Just what do you think if SEBA became more aggressive at promoting it? Would that change the way people perceive it in the professional community? Would it alienate them, or would you be happy to actually have some promotion of a product you like? By the way, this isn't going to be
off
published.
the record.
Yeah.